Hi, I'm Aldo Guzmán-Sáenz from IBM Research, and today I'll talk about uncovering signals of cell-state transition using topological data analysis in single-cell data. This is a collaboration with Can Risora Cry, Filippo Utro, and Lakshmi Parida. The development of effective treatments for drug-resistant cancers is running into limitations of bulk sequencing. One such limitation is that we cannot characterize individual cell states, and so cell state changes cannot be determined either. Single-cell sequencing allows for much finer resolution, but one of the new challenges it poses is working with much larger datasets. One of the tools we'll use is topological data analysis, which uses objects from algebraic topology to model combinatorial and topological properties of data. To help ground it, one way of looking at it is as a natural extension or generalization of graph theoretical tools to higher dimensions. We use Mapper, a TDA technique, in our pipeline to study relationships between clusters of cell samples and tease out information about transitions between cell states. Consider a point cloud like the one on the left. Assuming we have a covering, that is, a collection of subsets such that the point cloud is contained in the reunion, we can then construct a graph as follows. We add a vertex for each element of the covering, here represented with different colors in the middle figure, and for every non-empty intersection between pairs of them, indicated by red points, we will add an edge between their respective vertices. If we additionally have another scalar function that takes value on our original point cloud, we can visualize its values on the graph. We use PCA to reduce dimensionality of our data, keeping the necessary information to recover the original data with minimal loss. The linear nature of this projection aids when computing transition vectors. In this diagram, the idea is that even though the line is in a plane, we can still perform a lift of its projection to the original space. Here we have an overview of the entire pipeline. We start with a gene expression matrix. Then, after filtering and pre-processing, we have the original feature space, on which we perform a projection, PCA in our case, to a lower dimensional representation. We then apply a low distortion nonlinear projection to an even lower dimensional representation, where we run the Mapper procedure. Mapper yields a graph with connectivity information corresponding to transitions between cell states, and the final three steps are to lift such transitions to original feature space. We apply T2DA to a single-cell study of BRAF mutant melanoma cancer cells. This study consists of 674 sequenced cells categorized into 4 distinct transcriptional states according to MITF activity. T0 is pretreatment, phases 1 and 2 are transitory states, phase 3 is resistant state. We filtered out mitochondrial and ribosomal genes as well as MITF, which was used for labeling. To establish some baselines, we used UMAP to visualize cells and Leiden clustering to identify subgroups. We observed a heterogeneous mixture of cells, and the clusters don't seem to capture the different phase states cleanly. We also computed a PAGA graph of cell trajectories using the output from Leiden clustering. This trajectory analysis is highly interconnected and does not show distinction between cell phases. PAGA also can't identify the specific features that characterize the transition from one cluster to another. In terms of results, T2DA identified cell clusters and constructed a graph of related clusters presenting more homogeneous composition with respect to cell phase state change than ScanPy. We focus on edges connecting T0 predominant clusters to clusters predominantly comprised by phase 1 and phase 2 cells. We also focus on edges connecting phase 2 predominant clusters to phase 3 predominant clusters. Analyzing edges from T0 to phases 1 and 2, T2DA identified many potential features such as FOS, S100G, COM2, and MAGED2. Here we have listed the relation of these genes to melanoma. FOS, S100G, COM2, and MAGED2 were seen to have decreased expression moving from D0 to phase 2, which aligns with positive treatment response. Phase 2 to phase 3 transition captures the shift from a treatment-responsive to progressive state. Transition genes may reflect mechanisms of resistance that can be targeted. T2DA identified several genes associated to melanoma and other cancers, including S100A4, CTGF, CCND1, and IGSF8. Here again we have listed the relationship of these genes to melanoma and other cancers. So, to summarize, while T2DA projects to a lower dimensional space, it preserves a map to original feature space to make return to the original biological entity easier. When using T2DA with single-cell datasets with clear phenotypic states, such as disease staging or treatment response, T2DA can generate new targets for regulating or affecting the state transition. In this application, by looking at features that underlie transitions between T0 and phases 1 and 2, and transitions between phase 2 and phase 3, T2DA can identify genes that lead to a resistance state, and offers potential novel therapeutic targets for treatments that may avoid or overcome BRAF inhibitor resistance in melanoma. Thank you.